So the problem that I'd like to talk to you about today, and it's really something I'm passionate about. I've been thinking about this problem since I was a graduate student at Harvard. And that's the fundamental problem of how you extract genetic encoded information in our chromosomes, our so-called DNA, and how those molecular instructions are used and expressed. The term that we use is expressed. That is, how that genetic information is decoded to essentially make all of the molecules that keep us alive, cell by cell. So what I'm really interested in is, how is that process regulated? What's the molecular machinery that does it? And eventually really understand at sort of a temporal and dynamic way how that process works in a living cell. So why do we care about tracking biomolecules working in living cells? What I'm interested in is how that information that's locked in the DNA double-stranded molecule gets decoded and becomes information that makes all the molecules in your body, like the proteins that actually run the cellular functions of your body. This process is called gene expression. It's expressing the genetic information that's in the DNA. And the DNA is kind of fading there. It's in purple. And some of you will know that you have to copy the DNA into another molecule called RNA, and the RNA in turn is translated into a protein, and protein does all the work in the body. So this process that I'm trying to study, the decoding process, is called transcription. It's transcribing DNA into RNA. It makes a copy that's actually a complementary copy. And the proteins that do this are an amazing group of proteins called transcription factors. These are proteins that read DNA and make copies of it into RNA. And why do I care about this interesting biological molecule? Because these molecules pretty much control every facet of a living cell. So how you develop from an embryo to a full being, and every aspect of what a cell produces, how long the cell lives, how big the cell is, etc. And most importantly, really understanding the molecular machinery that does this job informs us about many, many aspects of disease, human disease, like cancer, like diabetes, like arthritis, Huntington's, heart disease, you name it. And with the recent advent of genomics, it's become even more clear-cut that understanding how you decode this information is fundamental and key to understanding human disease. So just remember, every cell in your body contains your DNA. It's the same DNA, but you notice the cells in your body are doing different things. Your liver cell is doing one thing, your brain cell is doing something, your skin cell is doing something, and yet they all carry the same genetic information. So how does that happen? How does a brain cell know it's supposed to be a brain cell? How does a liver cell know it's supposed to be a liver cell? And it all comes down to these transcription factors reading the right part of the genetic information to make a liver cell versus a brain cell. So it's what we call cellular differentiation. So you have embryonic stem cells that can become all kinds of different cells, like neuronal cells or muscle cells. And that decision is directed, or as I say, orchestrated by these transcription factors. And it's combinations of these proteins that pretty much make you who you are. So that I can't get any more fundamental in trying to understand a living system. Here's a kind of a more graphic way of thinking about it. The double-stranded DNA I'm just showing you here as a strip, green strip. There it is, kind of expanded in double helix version. And these proteins that I'm talking to you about, these transcription factors, have to search through your entire chromosome in the cell to find the right place to turn on the genes that are going to make a particular cell type, let's say red blood cells or liver cells or brain cells. And so what I'm really interested in is, how do these amazing transcription factor proteins do this? How can they find just the right place to turn on the right gene at the right time? And of course, if I don't do it right, you get disease. So why do I think this is such a challenging problem? Well, think about it this way. Every cell in your body, let's say it's this chicken egg here. And the DNA that you have in every cell in your body is the equivalent of a human hair that's 6 miles long that's been shoved into this egg. Now think about how you're going to scan that 6 miles in seconds time period to find just the right place to turn on just the right gene so that you don't keel over. So that's the trick. So can I measure this event? So the problem is a little like, where's Waldo? How are you, first of all, going to even see this molecule? And if it's zipping around really fast, how are we going to see it? And it's going to be in a really packed environment. The nucleus, which I just showed you, is this very densely packed DNA protein environment. So it's a little like trying to find your friend in a huge crowd. So you can't do it. And the problem for us is triply difficult because our naked eye can only see 10 to the third meter resolution and in about seconds time scale. What we need is to be able to watch something 3 orders of magnitude higher resolution, that's a billionth of a meter, and in millisecond time scale. So one trick that we can do is that we can make everybody that we're not interested in dark, and we can light up just those molecules that we care about. So it's kind of like telling all your friends to wear a fluorescent hat and then shine a light on it that only lights up the fluorescent hat and everybody else goes dark. So now you go from a very jam-packed to single spots of light. So that's one trick. The other trick, of course, is you take a movie, because we don't care about you just sitting there. We care about how you're moving. What does the motion look like? Having now learned something about the fundamentals of gene regulation, I'd like to present to you an interesting problem, which is that we're talking about biological macromolecules that are zipping around in the cell, in every cell in your body, and actually in every cell in everybody's body. How do we go about actually figuring out how it works? And this is something that confronts scientists all the time, is that even when you have the theoretical basis for asking the right question, you have to have the right solution, and that often involves technology. How do you measure these events? And so the question for you is, think about various ways that you might come up with for studying the process of gene regulation at the molecular level. Well, now that you've had a little chance to think about it, maybe discuss it with your friends and the teacher in the room, let me tell you a little bit about, both from a historical perspective and more recently, what exciting new technological tools we've been able to use to start to address some of the most interesting molecular mechanistic questions about gene regulation. Now, gene regulation is a very complex system involving many, many proteins. And so there are many tools that you could think of, and I'm sure some of you have thought about it, including using genetic tools, genomics tools, structural protein tools, electron microscopes, and so on. But let me just tell you one interesting solution that I've been particularly excited about, and is very, very new. One of the biggest challenges in understanding any macromolecular reaction that's going on in our bodies is that it's happening rather fast. In fact, the timing component, or what we call the temporal dynamics, is critical, because if something doesn't happen fast enough, we're going to die. So one thing is, how do we actually measure how fast the reaction is going, but not in a test tube, but what's actually going on in the living cell? We've been measuring reactions in the test tube for a long time, and we still do, and it's very useful, but it really doesn't answer the question of how fast or how does that reaction actually take place under physiological conditions in a living cell. And so recently, in our laboratories in Ashburn, Virginia, called the Genelia Farm Research Center, we've developed entirely new classes of microscopes. These are light microscopes, not electron microscopes. And the key is that with light microscopes, we can actually look at cells while they're alive doing their thing. And we've been able to figure out how literally to track the movement of molecules that are decoding the DNA as they are decoding the DNA. It almost seems like science fiction. And in fact, if you'd had asked me 5 years ago if that was possible, I would have said, not for a decade. And here we are. And so using what we call super resolution light microscopy, we can actually track individual molecules zipping around in the nucleus of a cell in real time. This is going to completely change the game because it will allow us to understand for the first time the kinetics and dynamics of the reaction. And this will have huge impact not only in our fundamental understanding of how gene regulation works, but it will have a major impact in our ability to design drugs and other interventions for diseases like cancer or diabetes or whatever disease you want to think of. Because in the end, we're really interested in all this stuff because we want to understand how disease works. But before we can understand how disease works, we have to understand how a normal, healthy cell works. And so that's why I'm really excited about this new methodology. So I'm sure some of you are wondering if you found that this topic of understanding mechanisms of gene expression interesting and maybe even a career. How do you get into it? Or for that matter, how did I get into it? Well, the first thing is I think many of us from a very young age, we're problem solvers. We like to solve puzzles. We like to figure out how things work. We like to take things apart. And sometimes we can't put them back together again, but the point is we're trying to tear things apart to figure out how they work. That's pretty much what I've been doing for the last 45 years of my life, trying to tear apart the molecular machinery. And the question is, how do you get to that stage where somebody's actually paying you to do this? And for me, it was, as often the case, kind of a windy road. You know, I started off in high school being really excited about mathematics because there was no molecular biology in high school back then when I went to school. And so I didn't know anything about what my ultimate career was going to be. But math really taught me to think in a very disciplined and quantitative fashion. And then later when I got to Berkeley, I very quickly realized that, yeah, I'm a reasonable mathematician, but there were students out there that were geniuses. And so I thought I better find something better and more suited for me to work on. And that's when I discovered first organic chemistry, and then molecular biology. And once I entered the world of molecular biology, it was like a kid in a candy store. In fact, I got so excited about it, I almost forgot about my regular coursework, and I would spend 40 hours a week in the lab. And so I think it's about passion. It's about, this is not a job. This is something you just really want to do, and we're just very lucky that we get to do it as our profession at the same time. And it's really about discovery. It's about the wonder of looking at something for the first time and knowing that you're the only human being on earth that knows that. And that now you have to take that and let other people understand what it is. And very often, you not only discover fundamental aspects of life, but you really realize that that can be really useful for people well beyond the scientific world. You know, how it's going to impact a devastating disease, or how you're going to save the world's food source, or how you're going to understand better what's happening in the oceans. And all of this is tied to understanding how a living cell works. And so I always feel like I'm working partly for my own curiosity, but largely for the rest of the world, because I think we've got to make our planet work better.